as a SNAB, where we make A-level biology as simple as a cell. So today we're understanding how the Hardy-Weinberg equation can be used to see whether a change in allele frequency is occurring in a population over time. You'll also look at understanding that reproductive isolation can lead to accumulation of different genetic information in populations, potentially leading to the formation of new species. So the Hardy-Weinberg principle is a null hypothesis used to determine whether a population is evolving. It predicts that allele frequencies will remain constant from generation to generation if certain conditions are met. So if observed data deviates from the Hardy-Weinberg prediction, then a change in allele frequency is occurring, indicating evolution. For the Hardy-Weinberg equations, so I would recommend you remember it. There are questions where they do give it to you, but it's just better off to remember, and it's just two set of questions. So P stands for the frequency of the dominant allele, Q is the frequency of the recessive allele, P squared is the frequency of the homozygous dominant genotype, 2pq is the frequency of the heterozygous genotype, and Q squared is the frequency of homozygous recessive genotype. Required for the Hardy-Weinberg equilibrium to hold true is that for allele frequencies to remain constant, the population must be large, random mating occurs, no mutation, no migration, and no natural selection. So using the Hardy-Weinberg to detect evolutionary change, so step one is use the observed data to calculate the allele frequencies. Calculate the expected genotype frequencies using the equation. So you just calculate the different values that you need. So calculate p squared, 2pq, q squared, whatever you need depending on the question. And then compare the expected frequencies with the observed frequencies. And if frequencies differ significantly, the population is not in equilibrium. And this indicates that allele frequencies are changing over time, so evolution is occurring. And so information you can derive from such calculations is that if it is in equilibrium, then no evolution has taken place. And if it's not in equilibrium, then one or more conditions were violated, such as natural selection, mutation, migration, or genetic drift. And it is highly recommended you do lots of questions on this topic because it's pretty definitive. It will come every single year. So just practice. So, reproductive isolation occurs when two populations of the same species can no longer interbreed to produce fertile offspring, and this prevents gene flow between populations. So, isolated populations can experience different mutations, different selection pressures, and genetic drift, and this causes the accumulation of different genetic information, such as allele frequencies diverging over time. So speciation is the formation of a new species. Again, you need to know the definition. And this occurs when reproductive isolation persists long enough for populations to accumulate genetic differences and become reproductively incompatible. So you have two types of speciation. So you have the allopatric speciation. So this is when the population is separated by a geographical barrier and different environments cause different selection pressures. And genetic divergence leads to speciation. So sympatric speciation is the second type of speciation. So the reproductive isolation develops even though populations live in the same area. And possible reasons are ecological isolation. So different groups exploit different niches within the same habitat. So examples include different food sources and living at different heights in vegetation. And this reduces mating between the groups. Then you have the temporal isolation, so populations breeding at different times. Another possible reason is behavioral isolation, so differences in courtship behavior. And the fourth one, this isn't really mentioned in books, but it's just good to know anyway, is polyploidy. So errors during the meiosis produce an extra set of chromosomes. So that means that they can no longer interbreed with the original population, and this can lead to a whole new species forming. So sympatric species and genetic divergence. So what causes the genetic divergence? So reproductive isolation prevents gene flow. So different selection pressures act on different subpopulations, and as a result, the allele frequencies diverge and reproductive incompatibility develops. So that is all from Professor SNAB today. Hope you found this video useful. If you have any questions, please let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.